So welcome back, everyone. The people are just flowing in from the waiting room, so we'll just give a few minutes for the numbers to come up. But the stream has started, so I won't sit here silently. So why don't we start? Actually, we'll start first with housekeeping, like we did yesterday. And just as a reminder, so the requirements for your course credits are to attend, I believe, at least 2 of the 3 sessions. And Marcello has just put the info in the chat there, which is the attendance code for today's session, which is 4. So as a reminder, you guys need to go to the website and register your attendance, in addition to doing the homework, which we'll talk about today, to get your credits. And I don't know how many of you guys are aiming for that, but you know, it's a really nice system that the SciNet folks have put together here for you know, allowing you to get some credit for attending these courses. Now, I would like to do another question and answer type round, opening up this session like we did yesterday. I appreciated everyone's fairly detailed introductions yesterday, and I don't need quite so much background from you there. But what I would like to know is how many of you folks are going to be planning on following along the session today live. So just to preface, we're going to be working in Python, running Jupyter Notebook, using code from a GitHub repo, which I gave you the link to yesterday, and I'll show you in a second. So it's Python environment stuff. So how many of you guys are planning on following through live? And what is the way in which you're planning on following through live? The ways that the options are, and I will go to the website in a second to actually articulate this in a little more detail. And you might change your mind, actually. But I just want to know what people are thinking at this point. The options are to use the SciNet Jupyter Hub, to use your local machine, which you may prefer if you're comfortable with working in Python and working in Python environments, or another option, for example, perhaps you are familiar and comfortable with using Google Colab for Python stuff, or whatever, something else that I haven't thought of. So if you guys can just put some comments in, like what your intention is in terms of following along here, and what environments you're planning on using. Good. Okay, lots of action coming in through there, lots of SciNets. Okay, some people on the machines, I kind of expected that. Good. So keep that coming in. Now I'm going to go over to, I'm gonna explain those options that I just described a little bit more, and indicate that I've actually written this out in some detail now on the README page for the course repo. So you can navigate to this URL, which, you know, I'll even put it in the chat, but you can probably see it in the, oh, excuse me, I need to share my screen, right? So don't want to make any assumption. Don't worry, you didn't miss much. But this is the time for me to share my screen. I'll share my entire screen. You can see everything. Cool. So as I was saying, this is the GitHub repo for the course. I had this in a slide yesterday, and I guess it should be on the course webpage as well, but it isn't yet. So sorry about that. But you're here, so you can see what I'm saying, and it will be. I will add it to the webpage, and I will put it in the chat. So find the chat. Here is the link. So go to this link, and there's lots of helpful stuff in the README. I will be going through the process of getting the code and running through it. So, you know, don't worry about kind of scanning through all of this and digesting it right now. We'll be going back and stepping through some of this process. But just to the first point that I mentioned just now, I have some instructions here for working on the SciNet Jupyter Hub. If you want to use Binder, which I actually haven't tested it for this one, but I've used it before, and I kind of pulled the stuff over and changed the requirements. So this should work, but on the other hand, it also could fail. But that's, Binder is great. Actually, I can tell you what Binder is in a second if people aren't familiar with it, and Google Colab, which I can just comment on as well in a second. But those are 3 options for following along, and there's notes on the README page about how you can do that in each of those options. Yeah, so I'll actually just talk briefly about Binder and Google Colab, and this is slightly extending past the, I guess, nominal theme of the SciNet course of stuff you can do on SciNet, but it's definitely within the spirit of computational science. So for people who don't know, so Binder is a tool. It's a resource that's provided by some charitable foundations. It's funded now by, but quite a small scale outfit. But what they do is, what it is it's a service that gives you access to a compute, what we call a compute, well, we just call it compute, but a server running in the cloud for a limited period of time with a limited amount of memory and disk space. Okay, so it couples really nicely to things like GitHub repos with small kind of executable examples that you want to try out and run through. Specifically, it uses Docker images, but you'll see on various GitHub pages Badger, that is just a link to this Binder service. So as I said, I'm possibly shooting myself in the foot by putting this here because I haven't tested it for some changes that I made, but I just want to kind of flag that to you if you haven't heard of Binder. It's a cool tool that you can use to quickly explore interactively the contents of a GitHub repo, particularly ones that are Python based, but definitely they also support R and Bash and C or anything that's kind of standard open source Linux operating system type stuff. It's a great way of just quickly exploring code without having to fire up your local Python server or log into a grid or something like that. It's just in the browser, new tab, bang, and then you're good to go. The second one here is Google Colab, and Google Colab is basically, for all intents and purposes, the same as Binder in all the ways I just described. It'll give you a small, actually, they'll give you more than Binder will, but they'll give you a server, a temporary server running in the cloud that will allow you to do things like execute Jupyter notebooks, run code, do various things. And I'll say as well, I'm actually quite interested to hear what other people's thoughts are about this. So actually, this will be a cool thing to have some comments in the chat on, and even from the SciNet guys as well. So my like working process as a scientist, right? So what do? Right? I lead a small team. We do computational neuroscience research that is partly analyzing brain imaging data and partly simulating brain imaging data with the type of models that we're talking about in this course. It's all, 99% computer based. And some of it is in more of a, let's call it a development mode. And some of it is more of a kind of throughput mode, if you like. So the development mode is building new things, writing new code, writing new simulations, new models, trying new things out, getting stuff working, right? And then once you have stuff working, you transition it often into away from, let's say, a small scale test kind of scenario in the context of simulations. I might be like doing a really short simulation or for a really small network or something like that, or analysis, like one subject out of 500. And then you transition it over to a larger context that often is going to be on a high performance computing environment like SciNet, where you need the beef. You need to be able to send out thousands of jobs and run, with high, large amounts of RAM and so on. And that's quite a common, I mean, it's extremely common. It is the way you do high performance computing, is you do small scale development and then you move over to HPC. But the reason why I'm going into this is that I have found over the last few years that to some extent Binder, but more so Google Colab, is quite a useful part of the, it fits into this process quite nicely, in that it gives me like a really rapid and kind of easy to access and particularly easy to share and collaborate development environment for testing stuff quickly. And when you kind of pull it in together with things like you can integrate with Google Drive, and you can use some of the things like folder sharing functionalities and Google Docs type things that you have with Google Drive. These things together, which I won't go into, the full details of my kind of productivity stack on that, but just to say like those things, they do gel quite nicely in that intermediate part of the scientific scientific computing development workflow of, it's kind of like transitioning over from small scale to large scale, and it has a kind of collaboration aspect in there as well. So I've found Colab like increasingly something that I'm kind of going to for certain small scale projects. It doesn't replace high performance computing environments and the kind of things you can do on SciNet, but I mean, in a sense, you could potentially be doing the small scale development on SciNet. That would be a common thing to do as well. But it's very nice for collaborative contexts and small scale projects. So I'd encourage you to look into it, do some Googling, do some reading, see if it kind of works for you. And it works nice for tutorials as well. So that's one of the reasons why I have the link here, and why to be honest, why I'm talking about it right now is when you're providing, running a tutorial or just demonstrating things to people in general, then you want to really minimize the assumed knowledge, but also assumed compute environment setup, let's say. The SciNet Summer School is quite nice in that they provide the Jupyter Hub and they provide a good solid environment for people to use. So that variability is less of an issue, but in other contexts where I'm doing teaching and trying to communicate things to people in a kind of teaching mode, there's not always something like a SciNet Jupyter Hub that we can use as the foundation, and it kind of defaults more to people's laptops or whatever the heterogeneous kind of architectures that people are working on in general. And that is an example when it's really nice to have Binder or Colab as either a primary or a kind of secondary environment that you can go to, you can point people to, and they can get going, and that it's going to work. That you're not going to spend half your time troubleshooting people's environment setup. And in general, with teaching, and Marcello and Ramses can speak to this, with computational teaching, like when we're doing workshops like this, the environment setup is really often it's half of the time that you spend with people is helping them get set up, which is really not the point, right? Really the point is to kind of be channeling the time and the kind of interactivity more towards productive learning around the content. So there are kind of good solutions coming through that allow tutorial sessions to be focused more on content than environment setup. Cool. So yeah, that was a kind of wordy introduction, but I hope that was maybe interesting to people, and they could share their thoughts on these issues and options as well. Please go ahead and use the chat and tell me, you know, how the world seems to you on these things, and you know, if you bad experiences and any things that I'm talking about, use information. Great. So let us think to the tutorial today. We're going to be going till 2 o'clock again. The plan is to run, well, in a second, we'll log into SciNet, into the Jupyter Hub, and we'll just take a few minutes to get ourselves set up there. And then we're going to run through a notebook, which I'll just point you to the contents of the GitHub repo here and orient you to what we're going to be looking at today. So you can see at the top level, we've got notebooks folder. The binder folder is just for the binder config. The data folder has some data in that isn't the primary thing that we're going to be looking at today. But I'll tell you what is in there relative to what it's used in a second. But the main stuff is in the notebooks folder, as well as the documentation details in the README. So there's a few notebooks in here. Today, we're going to be looking at this one. We're going to be focusing on this one, Intro to Whole Brain Modeling. And in the latter part, we'll be looking at the second one, which is a homework assignment, which follows directly on from Intro to Whole Brain Modeling. And well, we'll discuss this at the end of the session as well. What's also here, I'll just explain. So this one, exploring TVB connectivities and models. There's a little bit of redundancy here with respect to the Intro to Whole Brain Modeling, but it's also a useful thing to just flash through. And this last one, modeling resting state. Now I've included this in here. Audio is breaking a little bit. Okay, maybe I mean, I've had a few flags about a connection being unstable, which is a shame, but I can also try and stay. Yeah, please give me updates in the chat if there's any issues with sound and so on. Yeah, afraid I don't have a wired connection in this part of the room. Okay, so this final notebook, modeling resting state, this is another tutorial that actually I've used before, but I'm not going to use this time, but is definitely something to look at as you know, follow-on exploratory investigations if you're interested in the content. We're not going to be working through this tutorial here, partly because, well, mainly because it has some components in it that take a long time to run. Partly also there are some components in it that require a little bit more theoretical background than we've covered in this course. But definitely, if you're interested in this work, then the next thing for you to do is to open up this notebook and browse it, run through, and take a look at that. And what's done in this one, modeling resting state, is more of a focus on what we call functional connectivity or correlation patterns in resting state fMRI, and the way in which that's been approached in the computational modeling literature of the kind that that we're looking at here over the last 5-10 years or so. So reproducing some key results from some classic papers on modeling resting state fMRI. We're not going to do so much of that in this course. You know, one of the reasons is because modeling resting state fMRI requires you to do really long simulations, because you want to simulate, say, 10-20 minutes of data rather than what we'll be looking at here, which is more 500 to 1000 milliseconds of data. But I want to just to point to that and let you know that there's more good stuff for you here if you're interested in exploring it. But I won't say anything more about that today. So we're going to be focusing on Intro to Whole Brain Modeling. So let's do our login process. So as I said, I have listed the instruction set on the README here. I'll just talk through it and then we'll do it. So the first thing is you need to log into the Sign at Jupyter Hub. Now, hopefully everyone knows how to do that already. If you don't know how to do that already, there are instructions. In fact, I think I've explained this at the bottom here. If you go to the actual SciNet course webpage and log in, then there is a link that gives you login instructions for the Jupyter Hub. And specifically, it gives you a username and a password that you need to use. And those are not the same as your username and password for the SciNet website login. So if you haven't managed, if you haven't done that, I guess, one time over yet, then you're going to want to do that. So just do as I just described, but as I said, it's listed here, the process at the bottom of the screen to get the login details for Jupyter Hub. At this point, I'm going to assume that you guys have those login details, and we're just going to proceed to the Jupyter Hub login. And so we're going to log into the Jupyter Hub. Then we're going to do something that we have to do as a first time process only, which is to add the Conda environment that we're going to use to your local Conda environment list. And then we're going to clone the repo that I'm showing you the README from here. And then we're going to go into that repo and we're going to open up notebooks and we're going to run through the content. Okay. So here's the Jupyter Hub. Now I've already logged in. I'm not going to do the login for you because that would involve showing you my password, and this is streaming to YouTube. So I kind of deemed that something that I shouldn't do, even though it's only a temporary password. But this is the landing page that you end up at when this is the landing page that you end up at when you log in for the first time. What you will see is this folder Scratch. What you won't see is this folder here. And do I want to delete this? I don't think I have anything that I need in here, but I feel like I don't want to delete things on a whim. So I'm going to rename it so I can go through the process with you entirely. So pretend this isn't here, basically. You'll just be seeing a folder Scratch. If you guys at any point, you're trying to follow along and then you miss a step, or you're kind of a few steps behind, but you're trying to keep up, then just say so in the chat. And between me and Marcello and Ramses, we can definitely help you out with probably, we'll try and troubleshoot as far as like, time and everything else permits. Anyway, so you log in, you'll see this Scratch folder. Now, the first thing you need to do is, and now I'm going to just get some of these things out of the way. I'm going to minimize this. Don't need to see my face, really. So the first thing we need to do is do something in the terminal. So we'll go to the right-hand side, new. Okay. I want to keep the group chat open. I'm gonna have to just keep moving stuff around constantly. New terminal. Okay. So Jupyter Hub is great because it gives you Jupyter Notebooks and it gives you a Bash terminal. And what more do you need in life? So this is the Bash terminal. So the first thing we need to do is module load Intel Python 3. This is the there's loads of environment things that tells your bash terminal which Python executables to use. Don't really worry about the details of that, but basically what that then does is tells it to do things like look there for Python. And the next thing we need to do, I'm actually going to just copy it from the README page, conda config append. So you want to do the same thing. You want to just have the README page in the GitHub there, and just copy this line, conda config, copy this line over, conda config -- append envders /sinet /course SS2028 brain network env. And I've already done this, so fortunately, it's reassuring that I get a message telling me that it's already there. And that's added a conda environment to your conda environment list. So next thing, once you've done this, is refresh. Actually, go back over. We will use this terminal, so we'll leave the terminal open, but go back to home and then just refresh. And to check that this has worked, and this is kind of critical, right, is you go over to new again, and then from the notebook options, we should get this here. So this is giving us an option to create a notebook that is using the conda environment that we just added, which is called SS2020_TVB. Okay, so this is a conda environment that I created. A conda environment, if people aren't so familiar, is a set of Python like library installations, package installations that are grouped as a set and that can be loaded or are kind of initialized as a set in a way that's isolated from other installations, from the global installation or other environments that maybe have different packages and particularly different versions of some packages. So package environments allow you to have suites of packages that don't mess up other environments where you need different suites of packages. Okay, so you should get this thing on the right hand side here telling you that you have this environment available. We don't need to use it yet, but you need to know it needs to be there. Next thing is we're going to grab the GitHub repo. So we want to do this in the terminal. Now, as we saw, I already had it, but I renamed it, so I'm going to do it again. So this is just a Git clone. https://github.com, John Griffiths, that's me, and then sinet 2020 _ brain network modeling. Oh, I must have signed it SS2020, isn't it? Sinet SS2020. Yeah. Okay. No, nothing in the chat yet. So I'm gonna minimize the chat for now just to keep it out of the way. But we're getting some helpful additional help from the Sinet folks. We're all here to help you on the chat if you have issues at any point. Excuse me one second. Okay, so now we have our course repo, and this is going to be visible in our browser or our file tree here. So let's go in and go into notebooks. We see the notebooks here, the ones that I described that I was showing you in GitHub. Now, the way I want you to do this, and this is not, I think, universal, but I highly recommend, like this is a way of working with Jupyter notebooks. You grab a GitHub repo, a download folder, something like that. You have some Jupyter notebook that's maybe come from some package that you're interested in, some tutorial that you're interested in. it's tempting, and I think it's kind of normal to open up, excuse me, my nose wants to sneeze, but I'm trying to tell it not to. So the tempting thing to do, the normal thing to do perhaps, is to open up is to click on the notebook and open it up. Now, I want to suggest don't do that in general. Oh, excuse me. Bit of the hay fever at the moment, I think. Apologies for that. Okay, so don't do that. Don't open up the notebook. The reason not to do that is because then you start editing it, and if when it's something that you care about, it's documentation you want to follow, an example you want to follow, some work from your colleagues or something. Once you start editing it, and you start editing it, sorry, that was coming for a while. I'm really sorry to be peppering this tutorial with my sneezes. Okay, so this is what I want to say. Just duplicate the notebook and rename it. Okay, rename it something like whatever you like. You know, the default is copy, but you could say edited. Okay, and then work on this one. So you're not editing the original version, because you might want to go back to the original version. You might introduce some changes. Particularly, you might run through some cells and then find that, okay, I ran a cell that generated a crucial figure, and then I got an error, and then the figure's gone. And now, what do I do? I mean, you can go back to the GitHub repo or something like that, but it's kind of a messy way to do things. So this is how I recommend people work with Jupyter notebooks in general. If you're not the person who's developing that notebook, if it's not your thing, if you're looking at another thing, someone else's thing, then make a copy and work on the copy. Okay, so this is our notebook. We're going to start talking about content now. Hopefully the setup has gone okay for everyone. As I said, there are alternative options in the README. The Google Colab option probably should work well for people who are having any major technical problems. I will just quickly check the chat to see if we've got any concerns. Oh, thank you. Bless you. Yes, bless you. Bless me. Okay, no concerns from anyone in the chat. So I'm going to assume everyone's good. Now, so this notebook, it follows on from the tutorial that I gave you yesterday, and it works through some of the components of just setting up and running a neural mass model. Now, I'm gonna touch on it, just pop back and just give you a few quick reminders on a few of the slides, the kind of key slides that I had for you yesterday. I guess the first one is the overview. Okay, so what we're doing with whole brain modeling is we're using a reduced description neural population model. Our neural population models give us the basic units of a connectome network. They define the dynamics of the nodes in the network. The connectivity of the network we're taking from some combination of human diffusion MRI tractography and potentially also chemical track tracing data from primates. But bottom line is we get a connectivity matrix that defines our network, and then we simulate. That gives us time varying activity of various kinds, depending on exactly how we've generated the simulation. And then that gives us simulations of empirical data that we might want to measure. Okay. One of the key tools for using this, which is the one we're going to be using today, is a software library called the Virtual Brain. And The figure here is showing exactly the same thing as I described to you just now. But one of the things that is worth emphasizing here is that as we'll see as we're working through it, is these components of setting up the model are kind of modularized. Okay, so when we're setting up our brain network model, as we call it, we'll define a connectome, we'll define a neural population model. We have basically classes and objects that we define, that we initialize and define. And so we'll define a connectome, we'll define a neural population model. For some parts of it, we'll define a cortical surface. We'll define something that is meant by the signals here, which is basically the measurement model. And we kind of plug all these things together using TVB. What we're also going to look at here is the Janssen-Rit model, and I had a slide or two on that earlier on in the talk. So as a reminder, the Janssen-Rit model is a neural mass model that gives a summary description or reduced description of the organization of neural populations within a cortical column, as in within a a small slice of tissue that you would find in a small patch of cortex, and it describes the activity in that graph in terms of three neural populations: a population of pyramidal cells, a population of excitatory interneurons, also called spiny stellate cells, and a population of inhibitory interneurons. Okay, and one of the key things that we are interested in with the Janssen-Rit model is the generation of oscillations, which lead to resonance behavior, resonant characteristics that manifest as peaks in a power spectrum, and in the time series, they manifest as oscillatory waveforms. So we'll be seeing all these things in the next few minutes as we go through the tutorial. We also talked a little bit about these components of the individual populations in the neural mass model, which is the input and output operations, basically the synaptic kernel and the sigmoid function. We'll take a look at one of these as well along the way. And finally, one of the things that we'll wrap up with and that we will be looking at as the homework assignment is modeling brain stimulation. So taking our network, injecting a current or a stimulation burst, basically, into one of the nodes in the network, and then observing the propagation of that activity through the network, which is all very cool stuff, I have to say, my personal opinion. Biased personal opinion, of course. Okay, so we're going to go through the notebook. So this is just going to be me executing code and then commenting on the code. The notebook, everything runs really quickly. So I designed this notebook and this tutorial to be, you're not waiting around for a long time for anything. So I will actually execute the cells as I go. So there's some additional text that I've got in the notebook that's just, filling out the context, and you can definitely read some of this. I won't read verbatim the additional didactic text. I've covered pretty much all of it in the lecture yesterday, but I'll comment on a few things. But the first thing we want to do is set up. I have the way I organize my Python code, not everyone does it this way. I don't know if it's violating like the Python quote Pythonic way of doing things or not, but I like to do all my imports at the start and not be importing things halfway through all the time. It just feels cleaner, and it means you can have a section that's your setup section that like that's all the imports that you're going to need to do. That's all the libraries that you're going to need to have installed, and you're not shocked when you go halfway down, you're importing some library that you didn't know that you were going to need. So that's how I organize things. You'll see, people do things differently elsewhere. So we have our setup section. This first cell here, this is a nod to people who are using Google Colab, and it's worth commenting on here. So the two principal differences running conventional Jupyter and Google Colab. So Google, unfortunately, I definitely have to say, decided that they would make their own version of Jupyter notebooks that are almost the same, but not quite the same, and their own version of the Jupyter interface. So when you're in the Google Colab interface, it feels the same, but just to my mind, it's not as slick. It's really not as nice of an interface. It's a slightly new interface you have to learn. It's a real shame they decided to do that. They probably have their reasons. In fact, I'd be interested to know what their reasons were, if anyone knows. So yeah, but two things I want to say about Colab. One is the interface is slightly different. And secondly, the kind of mode of usage is slightly different as well, in that what you generally do with when you're using Google Colab is you do all of your installs within your notebook, rather than basically what we're doing here, which is having an environment that we initialize, and then the environment defines our installs. So I've included this cell here that excuse me. So I've included this at the top that is the kind of thing that you would need if you're using Jupyter notebooks, and if you're in Google Colab, you basically need to uncomment these lines and do these pip installs. And just in general, to be honest, it's not a bad idea when you're writing notebooks to have a cell like this anyway, even if your primary usage is not in Colab. But if you kind of anticipate it possibly being worthwhile, then it's not a big deal to just add a commented out cell here, and then for people, particularly people who are not super experienced, then you're helping them out to run something in the Google Colab environment. So I've got this here for people who want to do it. I didn't see anyone mention that in the chat, so maybe no one's going to use it, but it's there for you to add these non-standard Python library installations. Then everything should run in Colab. So first we'll just do our imports. Some standard Python imports, some visualization things, which I'll talk about in a little bit. And then TVB, the simulator platform, this is really the key things, and then some additional tools that we'll use for looking at power spectra. Okay, this one here it's a little bit messy, but it's a little auxiliary or utility function that I'm adding in that just gives me a slightly syntactically more minimal way of having this little code snippet here, which is used really often for inputting scalar values into TVB. Don't worry about it, but you'll see this kind of curious little function being called quite often, double underscore. I don't know if double underscore is a egregious Pythonic violation of a function definition. In fact, I'm interested to hear if anyone has any strong views on that. Software developers in general, they often have strong views on the syntactically right way to do things. So and it's good to hear those views as well. But it doesn't strike me as anything wrong with having a function that's called double underscore, albeit kind of curious. Okay, so working through. So this next thing is a rather long function that defines some matplotlib-based surface visualization. I won't go into the details of it, but we'll use it in a little bit. I have some explanation here about the neural mass model, including a little image snippet of the paper that was the original Janssen and Ritt paper. and a schematic of the model, which is, we actually saw this, I showed you a slide just now that kind of separates these three populations, and we have the wiring schematic here with the pyramidal cells driving the two interneuron populations, and then they're feeding back to the pyramidal cell population. The parameters are described here. I'm not gonna go through the parameters in the interest of time, but you can scan through to get a better feeling of what we're doing. So now we're going to start running some TVB stuff and doing some simulations. So the first thing we're going to do is initialize a model. As I said, things are modular. So in TVB, we have models, we have connectivities, and we have other things. Principally, models and connectivities are the main things that we're interested in scientifically. Oh, really sorry about that. So initialize our model. So we have a class, Janssen-RIT model. This has a number of attributes, such as the parameters. So these A's are synaptic connectivity parameters. I think we have things like, what am I looking for? Yeah, Numax is maximum firing rate. So the parameters are given as attributes that we can kind of explore in that way. And what I've got here is a little function that just runs what we call a single node simulation. So it's a single node as opposed to a network simulation, the network being the whole brain network. So I'm going to just take an isolated brain region and just generate activity for an isolated brain region. And there's a simple, basically minimal function in TVB that allows you to do called stationary trajectory. And then what I'm doing here is I'm just taking the outputs and putting it into a pandas data frame. And this is one of my biases in how I do things, is I generally stick things in pandas data frames as soon as possible because I just find them useful. So this is defining a function, and then this line here is just running it. As you see, it runs pretty much instantaneously. So we get back a data frame of neural activity for each of our 6 state variables. These being a current C and a voltage V for each of the 3 neural populations: a pyramidal cell, the excitatory interneuron, the inhibitory interneuron. Okay, so we have time series for each of these in our data frame. So we'll take a look at this. Now, one of the things, what I'm showing in the next figure here, looking at the time series, looking at this time series in 3 ways. And really what I'm highlighting here is something that we need to know about when we're running numerical simulations in general, in any domain, not just neuroscience, which is the role of initial conditions. And particularly, I mean, there are a few different roles that initial conditions play, but particularly here, a phenomenon called the initial gradient. So essentially what we've done in this simulation is we've given us some initial values for these variables, and the first, in this case, around about 100 milliseconds of simulation time has 100 milliseconds or so of simulation time it looks like a great big, we call it a transient, but a great big inflection in the time series, after which we see a more regular and, if you like, kind of normal looking behavior. So this initial transient, this is just, it results from the fact that we've set these initial conditions randomly. And the bottom line is you just need to ignore it. So you need to kind of cut out the initial transient. Sometimes it's got a burn in time. So the part that we're interested in this simulation is, well, I guess we would say maybe
250 milliseconds in, maybe we will be conservative and chop off the first 500 milliseconds. It varies depending on the parameters and the models, etc. So the second figure here, I'm just kind of showing you the transient in a little bit more detail, just zooming in on the first 500 milliseconds. And then the third figure here is just showing the simulation from 500 milliseconds onwards. So this is basically what we're interested in, is we've cut off our how do you say it, uninteresting kind of non-physiological, let's say, behavior at the start, and this is what we're interested in. So we have this oscillatory activity, and I was emphasizing oscillations before, and that's absolutely key for what we're interested in with brain rhythms and brain models in general. I just want to comment here as well. So, you know, it looks like we can see like clear oscillations in the excitatory inhibitory interneuron populations, but not in the pyramidal neuron population. This is just a scaling thing. So if we actually rescale the pyramidal neuron population by, for example, plotting it as a secondary axis. So in this case, we have a different scale for the blue line. Is it EIN? Okay, I think I got the axis labels wrong here. No, the colors. Sorry, the colors are different. The colors are different from this one to this one. So here, the green line is now what was the blue line in the figure above. So the pyramidal neuron population is being plotted with the y-axis scaling on the right-hand side, and the first two are being plotted with the y-axis scaling on the left-hand side. So they're actually, everything's oscillating, but just slightly different scales in terms of the magnitude. Okay, and we can see that also by just actually normalizing the time series. Now, next point of next section. So I'm just going to execute. Oh, I have a fire engine. It's going to be that a second. Coming to you from downtown Toronto, Bloor Young, where life is noisy. Life is wonderful, but life is also noisy. Okay, so next point in as we're going through is emphasizing one of the points that I was talking to you in the tutorial, which is that oscillations in these type of neural systems, particularly that have excitatory and inhibitory neural populations, are generated by negative feedback loops. So what I'm showing here is a set of simulations. I'm going to run this that are just varying some of the parameters that are the connectivity gain parameters between our neural populations. The actual parameters that correspond to these four entries, these four basically columns in this array, are given by these this kind of string title that I'm giving here. So it's the gain or the connection weight from the pyramidal cell population to the excitatory interneurons. This is the first one. Then the pyramidal cell to the inhibitory interneurons, the excitatory interneurons to the pyramidal, and the inhibitory interneurons to the pyramidal. Okay, and just to hammer that point home, if we go back up to our sketch of the wiring diagram, those are the four connections that we can see in this little diagram here. Okay. So we're playing around with the values of these, and particularly what we're doing is setting a number of these things to 0. So the number that you want to focus on here is really the which one is that I'm showing here? It's the 1, 2, 3, 4, 5th one. Okay. 5 and then 6 and 7. So if you look at the third column here, so the third column here is, as I said, the excitatory interneuron feeding onto the pyramidal neuron population. So we've got, actually we've got different values of these different parameters. What we're seeing in these figures here is the activity that we get when we set the gains or the connection weights in that little graph to 0 at various points, and then when we increase them, and the figure here in the middle is showing like the emergence of oscillations that we get when we allow there to be an inhibitory feedback within the circuit. So the inhibitory feedback that is coming through in the parameters for this simulation run is what's generating these oscillatory waveforms that we're seeing in this plot here. Okay, so next I'll just comment on this, but I don't want to kind of make too much of a point of it, because we'll focus on the things that follow this. Is that we in these models and how we think in terms of how these circuit functions work, the frequencies that are generated are influenced by the physiological parameters of the circuit. So this is just as a kind of demonstration that we can modify the connectivity parameters that we showed just now to generate different oscillation frequencies. So one example here, the right hand side is a power spectrum here. So if we just say look at this one, it's a slightly unusual plot because it's on a log scale. In fact, we're doing a log x. No, it's just a log scale on the y-axis, but not on the x-axis. But I want to point to here and here. So we have this damped oscillation here, but also the principal frequency of this damped oscillation is around 10 hertz. So we see a little bump in the power spectrum here. When you see a bump, that's what constitutes a rhythm, and then you get bumps for their harmonics as well in kind of pure sinusoidal-like oscillations. So we have a 10 hertz rhythm that we're generating here, but if we give some different values for the connectivity parameters, then we have principally a 5 hertz rhythm. So the first primary bump is around 5 hertz here. So no 5 hertz bump here in the one above. So there are other modifications we can do. I mean, these ones at the top are showing parameters that generate no oscillations, but we can also tweak or modify the actual frequency of the oscillations from the physiological parameters of the circuit. That's something that is probably a general principle of physiological organization, and it's the kind of thing that we can modify and study pharmacologically, amongst other things. So this next part, I'm just demonstrating the sigmoidal coupling function, which I described before. We're really just plotting something that's used within the model, but I had a slide on this, so I wanted to show you it again. So this is the y-axis is the output firing rate for a given value of the population average membrane potential. Don't want to focus on that though. So the next section, we're now thinking about the network. And we're thinking about the connectivity. So as I described before, we have our different modular components of the model, and we've looked at the model now, or the dynamics in the previous section. Now we're going to focus on the connectivity. So first thing we do is load a connectivity object, and we just this .from file method here is just loading the default connectivity, and then we have a little description in the HTML table here of what our connectivity actually is. So we've got 76 regions, 1,500 connections. It's a directed graph, as in it's not symmetric. So row to column is not the same as column to row. And there are other attributes that we don't need to think about here, but let's just take a look at what this thing is. So actually the next figure is doing a good job of it, but I'll just put it into a cell as well. So we have our con object. We want to explore it. We'll do dot and then just tab for tab complete. And this is showing us if you're wondering what I'm doing when I go off camera, I'm sneezing. I'm just trying to make it less obvious and less annoying for you guys. Okay, we have an error and a bit of troubleshoot. So I'm going to kind of try to come back on the error there in a second when I've finished this section. So just bear with me, guys. So we have our connectivity object. We can explore it. We just do dot and then tab complete. And the main thing they're interested in is the actual network structure, which is given by weight. So this is an array, just a NumPy array, 76 by 76. So that's the number of nodes in the network. And the next figure is just plotting that. Plotting two matrices that that are important when we're doing these simulations: the connectivity weights matrix, and the second one is the connectivity delays matrix, which gives the conduction delay between the regions. And then we're annotating it with some more connectivity object, which is the region labels, which are abbreviations that tell us what the brain regions are we're looking at. And just looking at these kind of figures, I stare at these kind of things all day. This is left hemisphere. This kind of block on the top left is one hemisphere. Sorry, the block is what we call intra-hemispheric connectivity, the connections between brain regions within the right hemisphere. And then the second block on the bottom right is the connections between brain regions within the left hemisphere. And then these little diagonal, we call secondary diagonal lines here. These are what we call the cross-hemispheric connections. So these are connections that connect left hemisphere regions to right hemisphere regions. And specifically, the only entries that are here in the off-diagonal blocks of this connectivity matrix are what correspond to the connections between homologous regions. So it's like left A1 to right A1. Well, actually, there isn't one there, but third one in left amygdala to right amygdala, etc. That's what this diagonal line that you're seeing and in the three diagonals is. Delays matrix is giving you a conduction delay corresponding to each of your connections. And then what we're doing in these next few sections is just looking at the brain regions that are the nodes of the network and the physical locations of them. So we have a set of XYZ coordinates for each of our brain regions. And this scatterplot here is just plotting it. They're kind of 3D XYZ coordinates, but we're seeing them in 3 planes. So people who know their brain anatomy broadly, the XY here is actually looking down from the top. It's an axial view. The X to Z here is looking from the side, so it's a sagittal view. And the XY here is looking through the middle, kind of back to front, or what we call a coronal view. And each of these dots is the centroid location of one of the brain regions in the parcellation or the connectivity that we're using. Great. And another way of looking at that, which is much nicer, and this is kind of what we focus on, is actually using this thing called the surface. The surface is a triangulated mesh of giving the shape of the cortical surface and the kind of location of points on the cortical surface a relatively high resolution. And we can use that to also visualize our brain regions with some, better brain anatomy. So each of these colored regions, colored zones, we could use probably we could find a better color scheme for this plot, but each of the different zones constitutes a different brain region. The function that I'm using to plot this is the one that's defined at the top, the kind of custom surface visualization function. And I have a little nod here that you can also do a similar thing using Nylearn's surface visualization functions. And the advantage of that is just import a Nylearn function rather than having a huge function definition like I have at the top there. There are some differences in how these things are plotted, but they're both using matplotlib, so they're not intended to be production value surface plots. Let's say they're just supposed to be functioning to a certain point. But each of these different colored zones is a brain region. And we can also use this visualization, take a column in the connectivity matrix and then plot the values of that column on the brain surface, and that will show you what the connection weights are for the brain region that corresponds to the column entry that we chose. In this case, so we're taking the first one, which is the right auditory, primary auditory cortex, A1, which is actually this little region here. So all of the red colored areas here, these are areas that have a connection to the right auditory cortex. Okay, so I'm just going to execute one or two of the things here that we're going to need. Okay, good. Now let's get to the simulations of our whole brain activity. So I've got a function definition here that is doing an extended version of what we did already at the single node level. And I'll just step briefly through the components. So we define the connectivity, which we looked at already. We loaded a connectivity, do an extra step here to just normalize the connectivity weights by their in degree. We have absolutely amazing time to develop a slight sneeze. So getting scowls from other people in the room right now. So we have our connectivity, we have our model, initializing our model as we saw earlier on. And then there's some additional parts of the simulation that we're going to specify here. We're going to drive the signal with noise, which basically means we're going to be using a stochastic integrations, numerical integration scheme. And that is something that generates more physiologically realistic activity, because a lot of physiological activity is probably driven by a background. You can kind of think of roughly as noise input level, although it's more like a background level of tonic input from other brain regions, but we can describe that as a noise term. So we define the noise, we define a stimulus, which we'll be talking a little bit more about in a second. We will define the functional form of the coupling between the regions, define our integration scheme, which is a stochastic scheme as I described just now, define some initial conditions, which just gives us more consistency in what we're seeing. And we define these things called monitors, which are transformations of the neural activity into we could call it a measurement model. So we generally call it forward model, but we call it a measurement model. So it's like converting the neural activity into something that you would actually measure, because you almost never measure anything that really corresponds to actual neural activity. When you measure EEG or LFP or something, it's not neural activity per se. It's not the state variables of the the physiological circuit. It's a physical signal that's generated by them. So we have a step that converts our activity into something more like a physical signal. We wrap these things all up into a simulator object, and then, as before, we run the simulation, and then we stick the outputs into some pandas data frames, and then we do a few extra things that we won't be focusing on too much here to look at the frequency content of the signal and then return the outputs. Okay, so we're just going to crack on through this and then execute. So this just runs the simulations for a certain set of parameters, and then we'll plot the results that we get. So in this case, instead of what we saw was a single trace, so we had our 6 state variables, and then we plotted each of those for one node in the network. Now we've got 76 nodes, so we've got 76 time series, and we're actually only looking at one of the state variables. We're actually focusing on the current of the pyramidal cells, which are the primary signal generating components of the network. They give us, they're the ones that generate the EEG signal, because they're large, highly oriented cells. So we have these, we're doing a time series plot and a power spectrum plot. The time series plot here, as you can see, there's a bunch of lines that are overlapping. So these are 76 lines. There's a line here for every node in our network. So we have a time series for every node in our network. You can see it's in a fairly synchronous regime at the moment because they're black in here and they're not completely random, but also not completely synchronized either. Our power spectrum is giving us the frequency content of this oscillatory signal. I mean, it's obviously an oscillatory signal, but basically the frequency, principal frequency is roughly 10 hertz, and this is what we how power spectrum looks, more or less how an EEG power spectrum looks. Actually, you don't see these harmonics quite so prominently, because you don't get as stereotyped oscillations. So we would maybe inject a little bit more noise into the system to get a power spectrum that looks, a time series that looks a little bit more like EEG. And then on the right-hand side is what we call functional connectivity, but specifically it's the Pearson correlation between each of these time series. And this is pretty interesting and pretty cool. So we've got, this is a matrix that's similar to the matrix that I plotted above, showing the actual connectivity graph structure. So it's, brain regions by brain regions. And then the entries are the correlation strength between brain regions. And we can see two main things, two things I would focus on in this. So it's just the correlation from roughly hilarious, roughly 1,000 milliseconds of simulation. And I want to just point you to two things. One is we have this kind of blocky structure. Now the blocky structure is in, we have roughly like a kind of a hot spot of red here on the top left, a hot spot of red on the bottom right. Let's have hot spots on the right and top and bottom left. This is kind of showing you that the intra-hemispheric regions, if you recall, the top left block is the right hemisphere. Actually, normally you would order things so that you have the left hemisphere at the top, but for some reason TVB wants to define it with the right hemisphere first. Doesn't matter so much, but anyway, within hemisphere correlations within the right hemisphere are relatively stronger, or relatively strong. That's what it means to see this hotspot here. And similarly, within hemisphere correlations within the left hemisphere are relatively strong. We have this prominent diagonal. The actual diagonal is kind of trivial because it's regions correlating with themselves. So, we would maybe zero that out. But we have like a kind of hot-ish penumbra around the principal diagonal. And very notably, we have these secondary diagonals in the correlation structure. Okay, so we saw the secondary diagonals in the connectivity matrix. In fact, they were the only things that you could see in the cross-hemispheric connections, the upper right and the bottom left parts of the anatomical connectivity matrix, or the wiring matrix. So that, what that basically indicates is that those anatomical cross-hemispheric connections are generating synchronous activity across the hemispheres. Which is also the kind of thing we see in your imaging data. So people who are familiar with looking at connectivity matrices in your imaging data, this plot here looks pretty similar to a typical type of functional connectivity that you see in your imaging data. So we're reproducing a qualitative feature of the type of things we measure empirically. Good. I'm going to skip the next bit, which is interesting, but the thing that we want to focus on finally. But I'm going to check back on the chat and see how people are doing with any technical problems. Okay, so firstly, maybe you figured it out already, but the person who's having the problem with the legend, just comment out the legend. It doesn't really matter if you're getting a legend plotting problem. The warning zip things, that's just a standard message. It's harmless. You're absolutely right. Is there a quantitative way to characterize and compare to actual data? Yes, 100%. We're not really doing that here. There is, so actually in the other notebook, which I described that we're not looking at today, there's lots of stuff on that. A typical way of quantifying for those things like these connectivity matrices, the fit to empirical data is actually just to correlate this matrix with a correlation matrix that you would measure from fMRI or from EEG or MEG. And then one of the things that I actually work on more is taking these things, these power spectra, from our simulations and looking at the similarity of this power spectrum function to what you would equivalent thing you would measure with EEG. And again, the same thing is, I mean, a Pearson correlation is not a bad similarity metric for a power spectrum. A chi-squared difference is also a objective function that's used sometimes. But yeah, for sure, like we want to generate these simulations and compare them to empirical data. We're not doing that in this tutorial because we're focusing more on just the components of the simulations. Okay, so final section is modeling brain stimulation. Now, so firstly, this attribute of the connectivity, the region labels, this is giving you, as you can imagine, like labels for the regions, like names for the brain regions. What we're going to do is we choose one of these regions. We're going to find the index that corresponds to it and just visualize it. So we can use the surfplots to just show us what is the brain region that we've picked out by just coloring it with 1 and the rest with 0. This is the left prefrontal cortex. We're going to inject a stimulus into the left prefrontal cortex. This is telling us something about our stimulus. So we're going to inject a brief square wave pulse after 1500 milliseconds of simulation time, okay, into this brain region. Good. So we have our simulation function. This is the same one we used before, but now we're actually doing the stimulation part and then giving it some information on the stimulation that we just defined above. And next thing is we're going to plot it. And these guys are really the highlight of the day, really, from the simulation results. So this is just a set of time slices plotting from, as I said, we're injecting it at 1500 milliseconds. So it's from just before when we inject the stimulation, and then immediately afterwards for the next 150 milliseconds or so. So first thing is we get high amplitude inflection or high amplitude activation of our stimulated region. And then we see over the next 100 milliseconds or so, our activity propagates out across the stimulated hemisphere. So that's plotting it as a time slice or a set of time slices. And then this next figure is doing the same thing. It's generating these surface plots, but it's doing them for a slightly finer scaling, and then using this tool called MoviePy to generate an animation of the stimulation, and kind of rendering it as a GIF. There, you can also render it using this method within MoviePy. Both of those is fine. The GIF doesn't work in Google Colab, but this is a really nice way of. It's actually quite a visually compact way of summarizing the result of our stimulation and seeing it propagate across the rest of the brain. Now, what is most definitely missing in this simulation, and this is it's an interesting scientific point, but it's also primarily it's more of a constraint of the components that we put into the stimulation. This simulation is that we're not getting propagation to the contralateral hemisphere here. We're only getting propagation within the stimulated hemisphere. Now, empirically, this simulation here, this corresponds to a type of data that we can collect. A very cool type of data, which is simultaneous EEG concurrent with TMS. So I described TMS to you before. TMS is a way of non-invasively stimulating the brain using a magnetic field. We can actually record EEG activity, which I just described yesterday, during, but particularly immediately after the injection of that stimulation pulse. And from the EEG, we can actually do what's called a source reconstruction to get at something that's more or less the activity pattern across the cortex that you're seeing here. So there is a type of data that is like roughly what we're simulating here is TMS, source localized TMS EEG, what we call a TMS evoked potential. And these are pretty useful experimental measurements for understanding what happens when you poke the brain magnetically in different locations with different characteristics of your stimulation frequency. So the final figure here is just plotting the same thing as a heat map, which is also quite a visually compact way of showing what we've got. So we have our stimulation region and then our primary stimulation point, and then we can see, if we had the nodes ordered, actually, particularly if we had the nodes ordered anterior to posterior, because it's a anterior region that we're stimulating, we would see really a kind of diagonal line all the way through, rather than these two diagonal lines in terms of the onset times of the other regions. But we can basically see, a gradually progressing activation across the rest of the regions, just shown as a heat map over time here. Great. So that's it for the demonstration. I'm going to go quickly now to the homework, and this is going to continue on from this final part of the tutorial that we looked at just now with the brain stimulation. So, notebook here, homework assignment. I'm going to not open that notebook. I'm going to open it in the GitHub. It really doesn't matter. So the assignment is described in the top here, but I'll just briefly go through it and give a few extra comments. So what I want you to do is take the simulations at the end of the last notebook, which is injecting this current into the brain region, and modify it in a way that you choose, or modify it in a few ways, try a few different things, and just summarize with some text and with some visualizations what it is that you're seeing. And you know, you can bring in some neuroscience knowledge here, or some neuroscience interests. For example, if you're interested in how does one of the things that you might do, and I would actually recommend a kind of straightforward way of addressing this challenge, is to choose a different brain region to stimulate, and then look at how the activity pattern propagating looks different for different stimulation sites. I have some other comments on how to do that as well. But other things you could do, depending on how much you want to think about it and how much you kind of understand the stuff that we've talked about so far, is changing some other parameters of the simulation and seeing how those affect the stimulation patterns. So 3 of those options here are the parameters of the neurophysiological model, which we looked at in the first half, things like the connectivity gains between the neural subpopulations, the conduction delays, and the connectivity structure, which is the weights in the network. So you can go in. I mean, the way you do this, you can go in and you can just manually change the numbers in the connectivity matrix. So if you're comfortable doing that, as you know, manually adjusting the connectivity matrix or the delays matrix, or uniformly scaling the connectivity matrix or the delays matrix, then those would be interesting things to explore in this homework if that's something that you're interested in doing. So the rest of this notebook is just giving you the starting point. So what I've done is I've just kept all of the cells from the other notebook that are necessary to run the simulations. A few of the figures repeated as well. And so you should be able to just execute everything up to here and then work from here. I've given a few extra comments that I've got in this section here about how to choose different region labels, so different stimulation sites. So the stimulation sites are defined. The way we're doing it here is we're just supplying one of the region labels from this list, okay, and then looking at what index that corresponds to, and then we can visualize it. So what you can do is, I mean, these are abbreviations, right? These aren't fully spelled out brain regions. So you'll need to of brain region parcellations, which you might be new to this. You'll need to find out what these regions are, right? AMIG is amygdala, okay. A1 is primary auditory cortex. You may kind of get the gist of this, but the way to find out what these region labels correspond to is firstly to just look up this thing called the regional map parcellation, which is where these abbreviations come from. So just a brief bit of Googling should get you that. But you can also just do it, manually, exploratorily, maybe combining with a little bit of insight into what you think the regions look like by plotting them like so. Okay, so I'm plotting a different one here, plotting left visual cortex, left V1, and that's another way of finding out where the stimulation sites are. Orbitofrontal cortex as well there. Okay, so so that's the plan. The format is to just run some simulations, change some things, generate some figures, write some text saying what it is that you're doing, why you're doing it, and what you find when you see what you'll find in the results, and just some commentary. And I'm looking forward to what you guys find. So I hope this is an enjoyable session for you. I hope this has been an enjoyable an enjoyable session for you. I'll just quickly scan on the chat to see if we have any final questions. We're just over time for the session, so we'll wrap up. So any final questions that people have in the chat before they get going with this homework? And this is due tomorrow, okay? Same time tomorrow, well, 2 p.m. tomorrow. And I'll start going through and looking through things after that time. And then in the session on Friday, we'll do some wrap up. I would very much like to have some, field some questions from you guys. They can be scientific questions. They can be highly procedural questions. They can be whatever you like. It'd be great to have a bit of dialogue in the wrap up session. I'll comment on some of the more interesting homework assignments. Yeah, like looking forward to maybe having a bit of discussion with you guys. Great. So getting a bit of responses in the chat. Thanks a lot. Okay, so have fun. See you guys on Friday.